Hi, this is Miss Litton, and this is my wonderful, wonderful APBio Period 1 class. Say good morning.
Good morning.
There we go. Okay, and we're going to discuss Investigation 5, Photosynthesis, the Sinking Leaf Disc Lab. Thank you, Max, for my golden pile of leaves. Perfect. Okay, Sabrina, can you go retrieve a hole puncher from your box of fun and a paper towel? Okay, so in this lab, you have your lab map to follow. Let's see if it links it. Go lab map. You have several links here that hopefully, tell your bio buddy, how many of those did you look at? Oh, no. Okay, now, all of those, like I said, can be helpful. Hopefully you worked on your background, and tell your bio buddy, how well did you do on your background from a scale of 1 to 10 answering those questions? I'm going to keep you 60%. You're going to do it for me. Okay, and today for your sign-off, you needed to be through the what? Data table. And then we'll talk about the analysis, claim evidence, reason, that part. But you want to take advantage of these things because they definitely, those links are going to help prep you for this lab. Let me get back to notebook. Where are you, notebook? Okay. So, okay, go away. There we go. You basically are going to take some leaves and you're going to infuse them with a solution. And you have some choice on that. The way the lab book sets it up is you infuse the leaves. What did the lab book say? You tell me. What were you supposed to do? Just what are the two solutions?
Water and soap, and bicarbonate.
Yes, water and soap, and baking soda or bicarbonate. Let me just give you a little tip. The ones that only get water don't flow. And the whole time you're running your lab, you're like, And yeah, spoiler alert, they just don't float at all. You might get them to float, but they don't float at all. So what ends up happening is you're just like 0, 0, 0, 0, 0. You may do that as a control. I have no problem with that, okay? Another option would be that you could make a 2% and a 4% solution, or a 3% and a 6% solution, and compare those two. Okay, that would be an option. You can assume I did the control, and with no baking soda, 0 floated. Okay, so that would be your control. Okay, but I just want you to be able to explore some other ideas as well. Now, you will have in your bucket of fun right here, okay, these are the things that you will have. Yes, some of the items are from Starbucks. I'm sure they don't mind. But you will have 2 beakers. I would label these 2 beakers. And you can label them. I put in some lab tape in here, and you can write on the lab tape with a Sharpie. You have a Sharpie in your bucket of fun, sorry, your pencil bucket of fun, and you put 2 labels on like I did, whatever your 2 solutions you decide to be. Oh, and you should clean them out in case they're dirty. Because this, when that comes into solution, whatever that is, could throw off your lab, right? So make sure they're clean and get rid of any kind of residual soap that's in here. So to make a true 2% solution, you would weigh out 2 grams of baking soda and do 98 mils then of water. We're going to make a little wiggle room here just to move the lab along. And I am okay if you use the beaker and use the 100 mils of water with your 2 grams of baking soda. It doesn't have to be exactly 98. You will still get the point. And I would put the baking soda in first, and then I would add the water to it. And the water that you need to use is distilled water. And I have to make this water, so don't clean everything with this water. I have to do a little rain dance and then hold up some leaves and gather it. No, I do have to make it though, so most of the water you should use is water from your sink, and you share a sink between you and another lab team. Use this water just for when you're running your experiments. As far as where the baking soda is, I've given each of you a little tube of baking soda that is here. And what you're going to do is you can use this to help, kind of put some out and put it in the cup. And where would we put this? On a what?
Balance.
Balance. Each of you at your lab station has a balance up in your cabinet up here. Okay? And so you want to carefully pull that balance down and you want to plug that in. And you don't need to use this. You can set that aside. Do you know how to zero out your balance? Yeah. Okay. So zero it out once you put your cup on, then put your baking soda on, pour that into your beaker, add in your water, and that's your your solution. Okay. Ask your bio buddy, are you good on that so far?
Are you good, bio?
Okay. Clarifying questions. All right. Now, in order to get your leaf discs, what you're going to do is this spinach, okay, get yourself a good leaf and avoid the veins the best you can. And it's kind of annoying because they'll all stick together. And you want to try to have 10 discs. When you run your two solutions, whatever you do, you want the same number of discs. If you can only get 8 sinking discs, then the other one has to have 8. And that's where this little tool from Starbucks comes into play that I've got in here. This is the disc remover, okay? And so it works great. And if you have some that aren't sinking, or this one has 10 sinking discs and this one has 8, remove the extra sinking disc with this, okay? And you see how this is set up like this? This is exactly how I want it when you turn it in. It might be a benefit for you to take a picture of it, so when you return it up here, it looks just like that. I am that specific. Those of you who know me, I want it to look exactly like the way I gave it to you, except the ones that are dirty. I want this clean. So you're going to take your disc, and over in your little, with all your pencils and stuff, you have a hole puncher. And I would, personally, I punch out like 13 discs for each one, so hopefully 10 sink. Does that make sense? And you're more likely, because some of them just won't. So you go ahead and punch out those discs for me, because I'll model how to do that in just a minute. Count out, they tend to stick together. You see how this is sticking together? You'll like pull them apart so you can see all, let's say, 13 discs. And you're going to put them into your syringe. Now, your syringe that you have, all you'll do is you'll just pop it out like this. And once count out 13, once you have 13, then put them in there. They'll tend to stick to your hand. That's just the way it's going to work, okay? And you'll put 13 discs inside of here. And I said 10, but you're more likely to get 10 if you put a little extra. Yes?
When I was reading through the lab guides of people who had done this lab, they said you should hole punch on the veins, but you want us to avoid your veins?
Somebody said on. No, wherever you have a vein, then you have xylem and phloem, you don't have the just chlorophyta. They should be saying avoid the veins. Yeah, that's what they should be saying. Okay, and then once you have your discs in there, this is the trickiest part, getting them to sink. But like I said, I think I have used a combination of things to master this, and I will model it for you. We'll see if I do it. But what you want to do is you want to push out all the air that you possibly can without smushing them. So I don't care how many you have right now, but go ahead and load them in here. I'll give you this. She did a really good job. So model Sabrina on that one. Okay, and what we're trying to do in this process is, if you didn't do this, there are all these air spaces inside the leaf. And so if you put them in a solution, then they would all rise to the top. So we've got to figure out a way to get the air out and the what? Yeah, the water in. Okay, that's what we're trying to do. We want to fill in those spaces. And then, though this is the very beginning of the school year and we haven't even discussed photosynthesis, you know plants do photosynthesis. You learned that last year. Okay, and what gas do they produce when they do photosynthesis? Oxygen. And as they do, the assumption is, as they do more photosynthesis, and as you produce more oxygen, that's going to fill the spaces and it'll make the leaves what? Float. Okay, so you could calculate the rate of photosynthesis by how fast they're floating there in your beaker, and you'll compare two different scenarios. Okay, perfect. Muy bueno. How many did you get in there?
I think 12.
Okay, hashtag awesome sauce. All right, so what you're going to do is you're going to take your syringe and you're going to push out most of the air. like that, okay? And then you will take a beaker. Okay, so you have beakers like this. And you'll have put in whatever amount of baking soda that you want. And then you'll add the water to it carefully. I'm just doing it quickly to show you. Are we all right so far? Okay, then this part's important. Don't make this sudsy. It's water with a little bit of soap, and it will help it infuse better, the solution go in. And I just want you to put in 2 drops. You can go. It says 1, put in 2. That's it. No mixing, no nothing. It's good to go. Okay? Now this doesn't have any baking soda in it, right? Right. So I just did that tomorrow. Okay? So then what you're going to do is you're going to go in and you're going to pull up approximately 5 mils of water. Sabrina, can I have your paper towel?
Yes.
And then you could do this next part over your sink, but I'm doing it here, so that's why. You want to get all the what? Air. And so I'm just going to kind of flick it like this so the air bubbles go to the top, and I'm going to push it till I get water coming out of there. We good? Now this is the part that hurts in just a second. Put your thumb right here, okay, and hold it like this. Okay, then what you're going to do is you're going to pull back on the plunger slowly, giving it a shimmy, shimmy, shake, shake, shimmy, shimmy. And you want to just coat them. You're trying to keep them in the water. Is what you're trying to do, and move them around a little bit, creating. Oh, I did not mean to do that. Let me see if I can rectify it. So then I'm pushing. I'm continuing to push up on my thumb. Do you see them sinking? And I pulled it out a little bit too far. Did you see the one sinking here on the bottom? So I lost a little bit. Okay, and you want to hopefully do it once. And so, but if you need to, pull again, and I'll be careful I don't pull too far this time. Learn from me. Okay, and then what I like to do is I push quickly up, and then just continue to push. You see how I got them all to sink? I just continue to push. Does it hurt my thumb a little bit? It does. Okay, but I'm okay. So now I can take these discs and I can go like this and open it right up into my solution. And I have a little bit in here, so I use my handy dandy Starbucks tool. Okay, and then I look to see, did all of them actually, oh, there's another one in there. Did all of them actually sink?
Yes.
You can't tell, but there's a few, there's 2 here on top. So I'm not going to use those. And I'm going to pull those 2 out. And then I'll count how many are down there. 1, 2, 3, 4, 5, 6, 7. I think there's an 8th one. It's stuck to it. Yes. Oh, 9, 10. I got 10 there on the bottom because 2 floated. I'm good to go. Okay. So that would be the process right here that you want to do. Okay. Ask your Bob buddy. Any clarifying questions on that? So walk me through this, and then I'll answer your question. Walk me through this so I know you've got it. Okay, your eyes are up here. You're not working on your lab prep because you forgot to do it last night. Your eyes are here. What do I do? What are you? Here I am. What do I do? I put the leaves in here, right? Okay. I make sure this water has my baking soda in it and my what? Soap. Okay, good. And then what do I do? Put the leaves in and then what?
Smash.
Okay, do I smash? No. Okay, yeah. And remember you're doing 2 of these at a time, right?
Yeah.
Okay, now what? Suck up the water,
5.
right? 5. And you can do even 6, it's fine. Okay, that part's not specific. Yes?
Did you want us to end with 5 milliliters after we push the air out?
It doesn't matter because you're going to put all the water back in here so you'll be good. Yeah. Okay, so I pull back 5 or 6 and then what do? Then I start shaking and pulling it down to the end, right? Okay, I get the air out. Then I shake, 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 shimmy, 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 shake. Then go quickly up and then just keep going and watch them sink. And then once you watch them sink, then you can remove your thumb. It goes away. It's all right. Okay? And then you're more likely to have really good success. Questions? Yes?
So we don't have to pull the plunger back approximately 10, like it doesn't matter?
That's what you suck it up. You suck up your original 5 like this, right? And then you're pulling it back to the 10 mark with your shimmy, shimmy, shake, shake, like this. And then you're going and continue until you see your leaf sink, okay? And then you can set them like this so they stay sunk. You know, you can set them right there. Okay, so I would recommend, because you have tape and you're labeling the 2 different cups, label your syringes as well, because there's nothing on it. You'd be surprised how often they get messed up, okay? So label your syringes as well to match your cup. And then when the discs have sunk to the base, dump them in the cup, and you're going to start your time. And every minute, you're going to check to see how many discs have floated. So you're just going to check on the minutes and say, oh, there's 3 discs, or there's 4 discs floated. And you have to decide what's a true float, because sometimes they're like part way up. You know, I would say they have to break the surface. But even when, you know, what's touching the top of the water, not halfway up the cup, but at that time, how many are truly at the top. Okay, and so you'll watch and you'll count the ones at the top. And remember, you're going to put a light on them. Now, I want to try some different lights out. So group 1, I already switched your light out to a different bulb. But my other students, and screw these on here like this. So it's up here. These get flipping hot. You will burn yourself. So this would be part of your safety you want to be careful of. Okay? These lamps are in between, and they're labeled 1, 2, 3, whatever lab station. This is lab station 1, that's lab station 2. So get them out from underneath. And in your box of fun right now, I have a better bulb, okay, that I think works better for this lab. Try not to put your hands on this. Anytime you put grease on a bulb, then that's an area where the light will be focused. You know, it can get hotter because of the oils. So try to touch this as little as possible. Take out the bulb I have in there, put it in here, and put this one in instead. And then I'll pick these up at the end when you put them back up here. Does that make sense? In team 1, I've already done yours. So add light to maximize the photosynthesis. You can put a heat sink over it if you want to, or try it without the heat sink. I did it without the heat sink, worked great. Yeah, like another container with water in it to absorb the heat. You're not going to be running it that long. I think you'll be okay without doing a heat sink on that. So you can vary the percentage of the baking soda, the distance from the light, you know, the concentrations of the baking soda, totally up to you. And then you're going to be recording the number of floating discs with water and just soap, and the other one with water, soap, and baking soda. And then I'm going to talk to you about this ET50 in a little bit. It's when half of them have floated that you put in there. It's important, do the same amount of discs in each, you know, do the same amount of discs. So however many sinkers, the one that has the least amount of sinkers, have that many sinkers in the other one. And then you can pull that out. And then we'll talk about the graph. I want you to be able to get the data, but focus on your quality. Okay, questions or concerns on the lab? Yes?
What is the cleanup first?
Okay, so the cleanup for the lab is you're going to call me over, you're going to show me your data, because I'm going to sign off on your rubric that I saw your data, and then I was good with it, okay? And you're going to show me your nice, well put together bucket, and then you'll put the bucket back up here. You know, I'll give you the sign off, and then you'll put the bucket back up here, and your lab station will look like it did before. I have some other setups at the lab station I want you to be aware of. You'll use these as well. This is probeware that you use with iPads. I have this set up for a fossil lab I'm going to be doing. So just don't mess with that. You know, if you're super interested, I can show you how to do it, but you don't need it for your lab, so just ignore that aspect. Within your lab stations are everything you need. The hole punches are here on the side. In this bucket, this is where you will find, for instance, when you're making your baking soda solution, you're going to want to stir it up really well. Your stirring rod will be in this clear bucket. I think that's all you need out of here, but that's where your whole, I think your whole punch is in there. Was it on the side? Yeah. Okay. But I might have pulled it. Yeah, in case it's not in here, it should be here. It would be in this box right here. Set this up so it doesn't break. You know where that, I think that's good. Any other questions or concerns? Okay. Don't take these to your stations. When you're ready to pour water, just come up and use them and then go back. And your leaves are here. Get a good one. Soap is here. find your lab station first and then come get your bucket. And be super smart.